Good day, dear ladies and gentlemen. This is our collaborative talk on space biology with Galina Nechitailo. This lecture is our view on the problem of biological life support system for future space mission. I mean the space mission to Mars and beyond, where the permanent support from Earth would be impossible. It is clear now that space flight factors influence on biological objects. How can we make space garden in such case? Can we transfer our experience on Earth to space garden? What knowledge on plants in space flight do we have today, after almost 60 years of human space flights? This is a large problem and cannot be properly overviewed in one short talk. I will focus on energy results, what were hidden for a long time due to different reasons, while other studies can be found in the literature. This is one reason. Another reason is that these studies are directed to the space garden as part of biological life support system for future long-term space missions, for example, or to Mars mission. As I know, there aren't other such massive and consistent biological studies in orbital spaceflight that were so focused on space gardening. Long space journey with a crew require a life support system to provide food, air, and water for crew members or humans. If journey is around the Earth or Moon, all the stuff can be provided from Earth, as it is working now for International Space Station. But if Mars or beyond mission, the provision from Earth would be unrealistic. We would need space garden for food production and for air and water regeneration. Such space garden must be successful for years, stable and productive, otherwise death of astronauts. There are two possibilities for successful space garden: to get harvest in space and regrow it again and again from seeds, or to take enough seeds to get harvest from Earth seeds every time. First way is more effective and make space journey unlimited, but this way is more complicated. Second way is viewed more robust, but amount of taken seeds limits the time of space mission. Both ways were considered from beginning of space era. For example, Tsiolkovsky considered space garden as a solution for long-term space missions. He wrote that humans and plants will be affected by unusual space factors such as microgravity. Another fact of space flights was discovered later, during stratospheric flights in 1930s about. This is cosmic rays. At that time, first small animals, insects and plants were sent to stratosphere on balloons. Later, the investigations were continued with sound rockets up to 500 km altitude. Real spaceflight investigations were started with orbital flights. First satellites were used for animal experiments. The first organism sent in its orbit on the board of second satellite was Laika, a dog. It survived. Third, fourth, and fifth satellites carried out a biological payload with organisms. Then a number of biological experiments were installed on manned and unmanned satellites and spaceships. Even first manned spaceflight of Yuri Gagarin carried out the Drosophila experiment. It was found that situation with living organisms in spaceflight is complicated, and it was clear that there is a strong influence of space environment on humans, animals, seeds, microorganisms, on everything what was grown on Earth and what was adapted to grow and develop on Earth. Space is different. However, short flights didn't give a real possibility to investigate the influence of spaceflight factors on biological systems. It needed a long-term flight experiments, as minimum as during a whole life cycle. New possibilities were opened with first long-time space missions. First vegetation of plants in space was done during 18 days orbital flight of Soyuz 9 spaceship. At this time in 1970, a group of biologists in Energia company was hired. Their task was to coordinate biological missions, design and install biological flight equipment, carry out biological experiments during space flight, and conduct after-flight investigations. As a goal was a space garden. This activity was a part of the space program for a long time. Then the systematic biological studies on Salyut and Mir stations became available. In this flight on Soyuz 9, the experiments with growing chlorella and seeds were installed. The first potato was growing in space. More possibilities appeared when first orbital space station, Salyut 1, was launched. Massive space biology program has been done during this flight. 23 days in station. That was first experiments with growing plants in space station. First artificial vegetation system on the board with watering, light and air flow for the growing plants. First green harvest from seeds was grown in space and cosmonauts tried to eat it. It was a great success. But there were also some alarming results. For example, the concentration of free radicals generated due to cosmic rays were found in stored seeds. You can see electron paramagnetic spectra of ground control seeds and space flight seeds. This is only 72 days in orbit, and this is low Earth orbit under radiation belts. This is clear result of different sensitivity of different seeds to cosmic radiation. First self-sufficient ecological system was tested in Soyuz 13 in 1973. Urobacteria produced hydrogen. Hydrogen-oxidizing bacteria eat hydrogen. It was a cycle. The system works stable on Earth and in space. In this flight, Chlamydomonadas proteus was growing in IFS-1 device and ROST-1 device. In 1975, the biological equipment was installed in Salyut 4 space station. A massive pool of biological experiments in space has been started. For example, the first experiment with fish in orbit was done at that time. You can see the Oasis 1M system for cultivation of plants in right corner. Such devices were permanently installed in the station on ground and tested before the launch. In flight, in orbit, only sets with substrate and seeds were changed during each mission. The devices provided fixed position of the plants in artificial substrate, regular watering, aeration, and illumination of the plants. At the same time, the devices provide safety of the biological experiments for crew and for other devices on the station. Unique and suitable equipment was designed, tested, and used such as Svetablock, Oasis, Phyton, Vazon for Salyut and Mir station. In this flight, for the first time, green onion was grown in space. First green onion were eaten by astronauts. And astronauts survived. First food grown and eaten in space flight. The microgravity was always a problem for the high plants. These first experiments with gravistat were done to provide the artificial gravity on the board of spacecraft. You can see section of first cone roots grown in biogravistat in the right bottom corner. Fungi were growing in Salyut 5 station. The different regimes of growing were investigated. Fungi showed different morphology. That was first fungi grown in space flight. Next station, Salyut 6, carried out a lot of biological experiments. For example, first experiments with Medusa set were done. You can see it in the right corner. This experiment is related to abiogenic synthesis outside of station in free space. The first experiments on origin of life. The cassette was exposed in free space for years. From station Salyut 6, the biological experiments were installed permanently and managed by permanent crew. First experiments with tissue culture were done in space flight. For these experiments, a line of space flight thermostats was designed. You can see it in right corner. Here you see cosmonauts Kovalyonok and Ivanchenkov with phyton device with onion grown in space flight. The photo has been done during communication with the flight center with biologists. The phyton device was designed in such a way that growing plant doesn't contact with atmosphere of the cabin. Seeds were outshoot from seeds gone into the substrate, where they started to germinate. Permanent crews were loaded with a long biological program as usual. For example, for the first time, electrostimulation of the plants in space flight was used. Artificial electrical field was applied during seed germination, root initiation, and plant growing. The device was compatible with electronic devices of the space station, and it was safe for crew and station operation. You can see it at the bottom, this device. For the first time, eggs were developed in space flight, and a lot of other interesting results with biogravity start to replace the gravity in other plants. In this flight, there were a wide number of experiments too. In particular, there were experiments to minimize the radiation damage effect with anti-mutagen agents. This is important to store seeds for a long time in space flight. Cosmic radiation causes radiation damages in seeds. Anti-mutagen agents successfully reduced this effect. Also, for the first time, Arabidopsis was blossoming in space flight. It was grown from seeds in phyton system. This flight was full with biological experiments. Here is a long list. Probably the most interesting are weed growing in our system. You can see it in the bottom left image. Electrostimulation of pea growing, biorhythm of Drosophila, and experiment in magnetogravistat. The last experiment is if a magnetic field can orient seedlings instead of gravity. Flags on board of Salyut and Mir station were growing 3-4 days in magnetic field gradient. The seeds grow along the field gradient direction. You can see seedling orientation in bottom stage, different versions. Next step is Salyut 7 space station. Cosmonauts Berezovoi and Lebedev, a permanent crew, have done a wide number of biological experiments. Electrostimulation of pea plants, leaded gravistat, microorganisms kinetic of growing, proteus growing kinetic, seeds exposure, orchids growing, abiogenic synthesis in free space. Point out, at the time, the microorganisms resistance to antibiotics was tested during the microgravity and found the increase of resistivity in space flight. This visiting expedition came in right time. The Arabidopsis gave first harvest in phyton system. It was the first experiment with growing plant from seeds in space, which gave seeds to seeds experiment. The possibility to get a harvest in space flight from seeds was shown for the first time. A path to long-term flights with biological life support system is open. Soyuz T-13 was a heavy flight. Cosmonauts repaired the station. which was frozen due to power problems. They restarted biological systems and long-term experiments again, because previous plants were frozen and dead. But when the station has come to work back, they did a large number of biological experiments. In particular, cotton was growing in space for the first time. The cotton grown in space had almost double length of fibers than the longest cotton fibers known on Earth. They started experiments with experimental cuvette with artificial substrates. It was important for optimization of plant cultivation. They started experiments on polymerization of acrylamide gel. The gel synthesized in space was used for protein separation in electrophoresis. The separation with space gel gave much higher resolution than usual technology with ground polymerized gel. New period of space biology has been started with Mir station. From first expedition, each crew carried out biological experiments on station. By volume of data, about half of experimental results were received from Mir station. These are genetic studies, morphological studies, harvesting first wheat, first birds from eggs, snails, cancers, first radish harvest, first cabbage harvest, first kidney beans, first tree in space. Tissue culture experiments gave increased biological activity of plants, first potato harvest, first result is long-term seeds exposure to space, 6 years, and got harvest from them. Mir station expeditions were more open for international scientific society, for collaboration. All these results can be found in the literature. These studies of high plants and unicellular organisms in space flight were aimed to make a space garden for harvesting in orbit. It was not so simple as it was seen from the beginning, and the main issue that was resolved in this course of biological research was the experimental confirmation of the possibility of growing and development of higher plants and other biological objects in the absence of gravity and the cosmic rays, absence of electric and magnetic fields, and in small closed volume of a cabin of spaceship and station. And as consequence of this, an assessment of possibility of creating and sustainable existing of the artificial ecological system, biological system of higher plants and unicellular organisms which can exist in space flight. It now appears that practically all factors of space flight have a strong effect on all living organisms in spacecraft. First, the flight itself has a mechanical effect. In all space flights, biological objects are exposed to overload and vibrations during the launch of the spacecraft. In this case, in the case of return of the spaceship to Earth, biological objects also experience vibrations and overloads. Secondly, in flight on modern spaceship and stations, microgravity affects a biological object, but complete weightlessness doesn't arise. A number of factors such as operation of various motors at the station, acceleration during docking, crew movement, inhomogeneity of residual Earth atmosphere, solar pressure, and other factors have an influence. They create micro-acceleration of biological objects. Third, in space, starting from near Earth orbit and farther in deep space, biological objects are exposed to cosmic rays and particles, high-energy particles. Radiation protection of biological objects at station, such as existing on Earth in natural form, doesn't seem adequately possible. Fourth, in flight, strong amplitude fluctuation in temperature, gas composition, and pressure may appear. Also, all modern ships and stations are equipped with thermostating and air regeneration system. Surprises and accidents cannot be completely excluded. Fifth, biological objects in flight are in unusual electromagnetic fields. The Earth's magnetic field is weakened in near Earth orbit and change over orbital tones. There is no electrical field like in Earth's atmosphere, but there are permanent and variable electromagnetic fields of the spacecraft's or station equipment. 6. The limited hermetic volume of the cabin of a spacecraft or station and their heavy saturation with equipment, made of various materials of course, and constant presence of humans create a unique gas composition of the internal atmosphere, significantly different from the Earth's atmosphere, both in composition of the gases and their concentrations, and the kinetic of changes in the parameters of the gas composition with time. The exchange of atmospheric gases and averaging of gas composition as it works on Earth doesn't occur here. 7. The limited hermetic volume of the cabin and presence of crew created a high concentration of specific microorganisms and fungi, which is not typical for Earth conditions. In spaceflight, the exchange and averaging of the composition of the microbiological environment in the spacecraft doesn't occur in the same way as it observed on Earth. All these listed factors are significant for growing, development, and reproduction of biological objects. Altogether, the spaceflight conditions are significantly different from Earth conditions. Therefore, it would be wrong to expect the absence of difference in organisms that grew and function on Earth and in space flight. Here is a full list of biological objects used and investigated in space flights. As you see, not all plants and veggies considered for biological life support systems in long-term space flight missions were tested in space orbital flights. For example, only wheat, cabbage, watercress, peas, beans, radish, potatoes, and onions were harvested in orbital space flights. Other plants such as rice, peanuts, soybeans, tufa, sweet potatoes, carrots, turnips, beets, sweet peppers, tomatoes, cucumbers, eggplant, and so on. They are considered as candidates for space garden, but they had not been studied at all or insufficiently studied in space flights without harvesting. Harvest for these crops in space flight wasn't obtained. To use these plants in space greenhouses, they must be investigated in space flight at all stages of ontogenesis up to harvest of the crops in flights. Research and projects didn't address the issue of growing fruit trees at all. Experiments with Limonia tree on board of Mir station have shown the possibility of growing a dwarf tree for minimum 2 years, but no flowers or fruits were obtained. The first experiments showed that tree can grow in space. These experiments have not been delivered or planned for the International Space Station or other space missions. Growing fruit trees such as lemons, apples, cherries, and others in flight can expand both the vitamin component and carbohydrate component of the food in space for space mission. The results obtained from studying the cultivation of plants in space flight showed that the biochemical composition and morphology of plants are sensitive to the factors of space flight. The cosmonauts ate the harvested crops on space stations starting from Salyut 1 station and didn't experience any trouble. while sometimes the organoleptic properties of grown plants in spaceflight were unusual for cosmonauts, and they were not so tasty. However, these are small portions, rather just for taste. The biomass grown in flight was not enough for main food for cosmonauts. It is necessary to create a space greenhouse sufficiently large where enough plants biomass will be grown so that biomass can grow in orbit, can be eaten by crew as a main dish for a long time. At the same time, analysis of organisms of the crew members can show what are organisms deviations that food grown in spaceflight can cause. The main result of these studies is the plants can grow in spaceflight. Space greenhouse can be available in general. Humans can be provided with food, water, and air in long-term flight. But the harvest in spaceflight can be expected as big as on Earth. Productivity of the plant in space garden will be lower than on Earth. Content of biologically active components such as proteins, lipids, sugars, and other would be different. More investigations are required, where such products grown in space can be used as main dish for the crew. These studies require coordination of all efforts on International Space Station, with aim to make a space garden as source for main food for astronauts. Then we can go further to deep space, Mars and other planets and stars. All these results in details can be found in our book published last year, if someone is interested.